All right, good morning, everybody. We're going to do something totally different in the second half of the course. So instead of talking about specific elements of the human body, we're actually going to talk about infectious agents this week. And then we'll talk about some of the wound healing processes that will go on that coincide with the infections. So let's start with something that's actually incredibly topical right now. We're going to talk and maybe spend a little bit more time than I normally would describing viruses. So what is a virus, first of all? A virus is a nucleic acid, so it could be DNA or RNA, surrounded by a coat of protein. So you can kind of imagine this shell of protein surrounding these DNA or RNA strands. Viruses cannot reproduce or carry out metabolic activities outside of a host cell. Viruses need to hijack your host cell machinery in order to replicate themselves. These viruses, and for that reason, are not considered to be alive. Viruses are important, obviously, and very pertinently because of their effects on human health, but I want you all to appreciate that they can be used as molecular biology tools to cure diseases, and there's some fascinating work going on right now with using viruses as a tool to ameliorate human health. So viruses pick their targets by specific receptors that are present on the surface of that target cell. And what types of cells and the number of species a virus is able to infect is referred to as its host range. So this is the number of species a virus can infect. And there's usually, like you can see, these proteinaceous glycoproteins that are embedded within the plasma membrane of the host cell. In this case, it's called CD4. These CD4 glycoproteins are able to bind to this particular virus here, and it's the attachment of the virus to the receptor embedded in the plasma membrane of the host cell that triggers the infection inside the cell. Viruses vary greatly with regards to host range. Some viruses can only affect one species, and in some cases, just one type of cell in that species. So like HIV, which is being shown here, only targets specific white blood cells, in particular helper T cells, because those are the cells that have those CD4 receptors on their surface. However, on the flip side of things, there's like West Nile virus, which can infect numbers of species, actually, mosquitoes, birds, horses, and humans. So there's a huge host range for that virus. A typical virus has a pretty predictable lifestyle that I'm going to break up into 5 steps, one of which is spelled wrong. So this is the life cycle, and in brief, the stages are attachment, entry, synthesis, assembly, and release. So in attachment, the virus, which is right here, needs to attach to some sort of protein or structure on the surface of the host cell. Once the virus attaches, that will allow it to enter the host cell. And you can see here the protein of the virus actually gets into the cell. But what we really are concerned about is the entry of the viral nucleic acid into the cell. Once you have viral nucleic acid in the cell, you can start this synthesis phase. So in the synthesis phase, you're going to use this viral DNA to A, make copies of itself, and B, get transcribed into mRNA that can then be taken advantage of by the host cell machinery, the ribosomes, to make viral proteins. And then you get to the 4th stage, which is now that you've got the viral DNA and you've got the viral proteins, you need to package them together so that they can form a full virus again. And this is called self-assembly of new virus particles. And then finally, now that we've made the virus inside the cell, we have to find a way of getting it out of the cell. And that's finally termed release. Each virus will have different variants of each of these steps, but in general, it's these 5 steps that are required for the virus to replicate. So just to remind you again, once we enter into the cell, our viral nucleic acid, which can be RNA or DNA, so they're just going to be a little strand of nucleic acid. They could be single-stranded like I've drawn there, but they could be double-stranded as well. So whatever the case, once you get your viral nucleic acid into the cytoplasm of the target cell, you can then transcribe and translate it into all the different virus proteins. And you can use DNA synthesis or some other method to make more copies of the viral DNA. And then now that you've got the viral nucleic acid and you've got the viral proteins, they can basically more or less spontaneously assemble into new viruses. And then, of course, you need the virus to exit the cell. I'd like to spend a little bit of time talking about the HIV virus in general, because that's something that's described this week. So these red ribbons inside the virus are the RNA single-stranded genome of the virus. So there's 2 strands of RNA that are packaged within this virus that are responsible for encoding all of the proteins in the virus and basically determining the behavior of the virus. The capsid, and this is a word that we haven't talked about yet, a capsid is the protein shell that's surrounding the virus. So the capsid in HIV looks like this, almost like a coffin. And then there's this outer ring of proteins as well. This is also an enveloped virus. And enveloped viruses have taken some of the plasma membrane of the host cell and actually are wrapped themselves in that plasma membrane. So you can see the HIV virus, in addition to be surrounded by these capsid proteins, are also surrounded by a plasma membrane as well. And embedded within the plasma membrane are these sort of pink pillar-like proteins that are called GP120 and GP41. And these are going to be involved in the attachment stage to the CD4 receptors that we talked about earlier. Also packaged within the capsid are tRNA, so you can help to some extent translate the RNA sequences. We also have an enzyme called protease, which is going to help digest proteins in ways that we'll see later. We also have an enzyme called integrase, which is going to help insert the viral genome into our host cells. And then we also have an enzyme packaged with the virus called reverse transcriptase, which is going to take this single-stranded RNA and actually convert it into double-stranded DNA. So that's the HIV virus. Again, we've got these proteins that kind of form the shell that are called the capsid. Within the capsid, we have our genome, 2 strands of single-stranded RNA, packaged with protease, integrase, reverse transcriptase, and then the whole thing is surrounded by a viral envelope in which are embedded the viral proteins GP120 and GP41. So here's the replicative cycle of HIV, and remember that there's several stages that we need to follow here. We first need to see the attachment of the virus to the host cell. We then need to see the viral nucleic acid entering into the host cell. We then need to see synthesis of the viral protein and nucleic acid. Those will then reassemble. And then finally exit the cell. Okay. So what I'd like to show you here first, of course, is the attachment phase. And this is due to those GP120 and GP41 proteins in the viral envelope binding to the CD4 receptors on helper T cells in the human body. The attachment of the virus to the host cell will allow for entry to occur simply by the fusion of the viral envelope with the plasma membrane. So here you can see the fusion has taken place. And then this will essentially deposit the viral single-stranded RNA into the host cell cytoplasm. So the capsid then releases its single-stranded RNA, and then that first enzyme in the capsid becomes active. That's called reverse transcriptase. And what I said, reverse transcriptase takes our single-stranded RNA of the virus genome and converts it into double-stranded DNA. Remember, what's in our nucleus is double-stranded DNA as well. And so now that integrase protein that's also packaged with the virus is going to essentially cut and paste the viral double-stranded DNA into our chromosomes. So now the viral DNA is part of the chromosome of the cell, and as the cell replicates and divides and makes copies of its nucleic acids, it will also make copies of the viral nucleic acid. So our viral DNA is now in the host DNA, so this is a pretty insidious system. Now we get into the synthesis stage. So if you've got host DNA, you can simply transcribe it into mRNA, which can then get converted into various proteins. Which proteins? Well, integrase, reverse transcriptase, protease, GP120, GP141. All of those are going to be transcribed and then translated from the DNA that's embedded now in the host chromosome. You can also use DNA synthesis to generate more of the genome RNA. And now, since you have your viral proteins and your viral RNA, we can simply package them. In the case of HIV, the proteins and the nucleic acid do not reassemble inside the cytoplasm. Instead, they get packaged into a vesicle that gets sent out of the cell, and it's within those vesicles that the viral particle will then mature. And what I mean by mature is that the protease enzyme is going to cleave the proteins that were synthesized in the cell into their functional components, and the virus will actually finish making itself when it's outside of the host cell. So those are the different stages of the viral replicative process. Here you can see the review of the whole process in general, but again, we have our HIV virus. The GP120 and GP41 receptors of the virus bind to the CD4 receptors of the helper T cell. That causes the fusion of the viral envelope, and the virus now enters into the cytoplasm where it releases its ssRNA genome. Reverse transcriptase will then convert that ssRNA genome into a double-stranded DNA sequence, which the viral integrase protein will then insert into host DNA. Because it's in the host DNA, it can be transcribed to make mRNA or transcribed to make genomic RNA. The mRNA will then get translated into viral proteins. The genomic RNA that gets transcribed from the host DNA is now made as well. These are going to come together, get packaged into a vesicle that's budding off of the cell, and the virus, based on the protease activity, will rearrange and mature when it has exited the cell. So what you see is during the course of AIDS, and AIDS is a syndrome, HIV is the virus that causes it, we see a number of different steps. So we start off with, at the initial stages of the infection, when you become infected with HIV, you have some general sort of classical flu-like responses: fever, fatigue, weight loss, diarrhea, aches and pains, just your body's kind of aching. This is referred to as the primary infection. So it's the first time you actually get infected and your body's starting to respond to it. And we're looking at 3 different measures of things. In red here is the amount of HIV in your blood. So you can see when you first get infected, there's this massive spike of HIV particles. In blue is your antibody load, so how you're going to fight the virus off. And notice again, you start low, but then eventually you start increasing your levels in blue here. And at the start of the infection, your helper T cells in green are at its normal healthy levels, but they're going to drop because the virus is going to kill them. At that point, we enter into our second stage, in which the immune system kicks in. It starts to produce antibodies, and the number of free viruses drops. So you can see in blue, the number of antibodies is rising. In red, the number of virus is dropping because the antibodies are doing their job. However, because the virus kills helper T cells, our helper T cells levels have dropped, but because the virus is dying off, we're starting to see our helper T cells recover. So in this stage 2, 1 billion viruses are killed today, but eventually so are 100 million T cells. There's no symptoms at this point because the war has just begun. So what happens over many years, in some cases, many weeks or months, depending on the strain of the virus that you're looking at, the integrated viruses will continue to replicate and kill helper T cells, but our antibodies will continue to target the virus and kill the virus. And you end up with this kind of long-term tug-of-war between those two things. You need the helper T cells to generate the attack against the virus, but the virus attacks the helper T cells. So this is the tug-of-war that happens. So over 3 here, what happens around 3 is that the integrated viruses continue to replicate, and enough viruses are not able to make enough helper T cells. And then you get to stage 4. Eventually you reach some sort of threshold where the number of T cells has become so low that you're not able to generate an immune response, and now the virus is starting to creep up, and the antibody levels are dropping along with the helper T cell levels. So the virus is now beginning to almost exponentially explode in terms of population, and again coinciding with the sort of drop of antibodies and helper T cells to go along with that. And at some point here, you reach 5, which is where the immune system is just totally compromised, and now all these other harmful diseases, not the HIV virus itself, but other normally harmless diseases that would not cause any trouble to a person that doesn't have HIV/AIDS, will now have a bunch of things wrong with them. You know, colds could now kill you. You may be exposed to certain bacterial infections that normally wouldn't infect a healthy person, but can infect an HIV/AIDS patient. And so at this point, the patient is eventually has lost the battle, and the virus will replicate out of control. Their helper T cells will drop to 0. Their antibody levels are going to drop to 0, and they'll get killed by any number of usually harmless infections that are just floating around, that are infecting us all the time, but don't do any damage to us. So we call the sort of extended stage clinical latency, and then we call the diseases that sort of take advantage of the fact your immune system is compromised as opportunistic diseases. And then finally, the end of this, unfortunately, is death. And I just want to emphasize that AIDS is not a disease. It is a syndrome, in other words, a group of diseases that is currently defined as the presence of several opportunistic infections. So again, these are things that don't make healthy people sick, but seem to make people with end-stage HIV/AIDS sick. We also need to see in the blood of the people with this syndrome the presence of interferon induced by the HIV virus. That will mean more to us in a bit. We'd also expect to see very low numbers of helper T cells, and when we scan the blood of the patient, they should also test positive for HIV. So in order to have AIDS, you need to have all of these 4 things. So how do we kill the disease? Or how do we kill the infectious agent that is causing the disease? Well, there are a number of mechanisms of antimicrobial action. This is a mistake on the slide here. We're trying to actually kill the virus or the bacteria, so we're going to use antimicrobial action. So one of the ways that you can kill a bacterium, for example, is by destroying its cell wall. And this includes a lot of very famous antibiotics like penicillin, cephalosporin, vancomycin are ones that I'm quite familiar with. The idea here is if you destroy the bacterial cell wall, you make the bacterium really susceptible to osmotic stresses, and the cells will either shrivel up or burst. We also can target the unique elements of protein synthesis in a pathogen. So these are the antibiotics like aminoglycosides, tetracyclines, chloramphenicol, and macrolides. We also can target the unique membranes of the microorganism. So this could include polymyxins, for example, and polyenes if you're using antifungal agents. There may be metabolic pathways that are unique to that particular pathogen, and you can target those specifically too. Sulfonamides, for example, do that. You can target the specifics of DNA or RNA synthesis in these structures. So actinomycin works in that as an antibiotic. Nucleotide analogs are often used to fight viral infections and target the unique elements of viral DNA or RNA synthesis. And you could actually just deal with the start and stop the virus at its first stage of infection, which is, remember, the attachment phase. So if you just stop the virus from being able to attach to the host cell, you have in effect eliminated the viral infection. So the problem with this, particularly when we talk about protein synthesis, cell membrane disruption, metabolic pathways, DNA and RNA synthesis, is that in these cases, humans have the same thing as the pathogens. And that leads me to something called spectrum of action. What you want when you're using a antimicrobial drug, like an antibiotic, is you want the drug to be able to specifically target a feature unique to the pathogen that isn't present within the host. Like, for example, we have ribosomes in our body. Bacteria have ribosomes in their cell. If we used an antibiotic that destroyed ribosomes, it's true, it would successfully kill the microorganism that's infecting us, but it would also kill us because we need those ribosomes to live. So when you're looking and designing drugs and you're trying to choose which drug to use, you would prefer to use a drug that is very specific to one type of bacteria that you're trying to destroy. So quite literally, the spectrum of action is the range of organisms a particular antimicrobial drug can affect. There are narrow spectrum drugs, which only affect a small number of species or even one type of species. There are broad spectrum drugs that can affect a wide range of species. These broad spectrum drugs can be, in some cases, quite dangerous because, remember, throughout our body, there are these microbial antagonists, these natural flora in our body that we talked about in Biology 1160. They're living all over our body, and they're working with us in a symbiotic relationship. They're not harming us. In some cases, they're actually benefiting us, and they are definitely not harmful to us in the position that they're found, but generally they're good for our body. If you were to take a broad spectrum drug, you might be endangering the cells of your own body because you may have enough in common with the bacteria you're trying to destroy that the drug will also hurt you as well. But much more likely is the drug is going to destroy some of the natural flora on your body, which can leave you vulnerable for opportunistic infections. So, for example, we have, say, E. coli in our large intestine. That's actually quite important. It's essential for helping produce vitamins, to help us absorb calcium in the digestive tract. So this E. coli in the large intestine is very useful and helpful to us, and we want it to stay there. Its presence there is stopping other bad things from being able to enter in there. So if we were to take an antibiotic at the hospital for some other infection, and it were to accidentally or inadvertently also kill the E. coli in our large intestine, now we have an empty space in our large intestine, and any old bacteria that might be present in the hospital room or in your body already somewhere else might move in and occupy that territory. And one of the most common opportunistic infections that can replace E. coli in the large intestine is Clostridium difficile, or C. diff, which you probably have heard of, and is a very, very relevant and significant infection that takes place largely in hospitals. But as another example of how about how E. coli can go wrong, if you were to somehow eat E. coli and it suddenly became access to your, say, esophagus or stomach or small intestine where it's not supposed to be, E. coli can make you sick there. So even if something is good in one part of your body doesn't mean it's good somewhere else. Another example of this is like yeast infections, for example, which often are treated with tetracyclines or erythromycins. The antibiotics will not just kill off the yeast that's causing the infection, it will also kill off the normal biota of the vagina, but not the yeast that are around there. And that's why the yeast infections can happen after a tetracycline or erythromycin infection. What you're looking at here is some very common drugs, and you can see what their range of action is. So in viruses, most of the spectrum of action just is limited to viruses, so they don't cause much trouble to us. You can look at various bacterial antibiotics, polymyxins, penicillins, streptomycins, erythromycins, tetracyclines, sulfonamides. You've probably heard of most of these. So notice the polymyxins are very specific to gram-negative bacteria. Penicillin kind of is mostly gram-positive, but also affects some gram-negative. These erythromycins tend to affect a lot of species of bacteria. And then these sulfa drugs could be really dangerous because in addition to damaging prokaryotes, they also damage eukaryotes. And don't forget, we are eukaryotes. And so if you start to damage eukaryotes, it's much more likely that you're also going to damage humans. And this leads to a very significant challenge that people face in the medical profession, is that sometimes giving the patient the drug to treat the infection will hurt the patient more than just letting the infection run its course. It really just depends on the circumstance. Another thing that you've probably heard of before is antibiotic resistance. So let's say we have a bacteria right here. The bacteria will multiply by the billions, and so now you've got a buttload of bacterial cells. Because of the nature of the replication process, there will be mutations or mistakes in the copying of the nucleic acid. It's not 100% copy from one generation to the next. And so some very small number of those billions of bacteria that have been produced will carry a mutation that will make the bacterium drug resistant. So in a population of, say, 1 billion bacteria, you may have 1 or 2 cells that are to some extent resistant to the drug. But when you expose the bacterial population to the drug, what's going to happen? Well, the cells that don't have any resistance to the drug are going to die very quickly, like the drug intended. But the cells that are resistant to the drug will take longer to die, which means that they're going to divide more than the cells that died faster. And in the next generation, you're going to see a lot more of the cells within the bacterial population carrying the resistance. And if this can go on for a number of generations, you can just end up with very drug-resistant bacteria. In fact, there are these extreme drug-resistant strains now present in, say, South Africa, where I think there's something like 5 to 6 major antibiotics will not work. The bacteria is already resistant to it. These resistant bacteria populations tend to be found in hospitals because that's where we're treating patients and the bacteria with the drugs. And so this is a pretty important story, and I'd like you to make sure that you're comfortable with it. The reason the bacterial population becomes resistant is because there already exists within the bacterial population some degree of resistance. And when you expose the bacterial population to the drug, those cells that are slightly or fully resistant to the drug are going to replicate more than those that aren't. So that when we look in subsequent generations of this bacterial population, there will be a much greater proportion of cells that carry the resistance. This is classic natural selection for those of you that are interested in continuing on in biology. This will become a pretty big topic. Okay, after this, I'm going to just give a brief overview of the innate immune systems and just how the immune system works in general. I'll see you then.